Welcome to the deep dive. Let's start by thinking about something absolutely fundamental to life, but maybe something we often take for granted. Timing. Not the timing of this conversation, but the incredible, precise internal clock that guides a developing organism. How does a cell know exactly when to perform a specific task or, switch to a new developmental program?
It's one of the oldest and most fascinating puzzles in developmental biology, really. Think about it. A single fertilized egg has to divide, differentiate, form complex structures.
All in sequence.
Exactly, all perfectly choreographed. If that timing is off even slightly, the whole process can just, well, go wrong.
And to explore how this timing works at a molecular level, we're diving into a classic system that's taught us so much. The tiny nematode worm, Caenorhabditis elegans.
Those elegans.
The elegans for short. Scientists just adore this little worm because its development is remarkably predictable, almost like clockwork, cell by cell, stage by stage.
Right.
Which makes it a perfect model to dissect these intricate timing mechanisms.
And within C. elegans, researchers discovered a special class of genes they called heterochronic genes. These aren't your typical genes building structures or doing metabolism. Their primary job is specifically to control the timing and sequence of developmental events after the worm hatches.
So post-embryonic timing.
Precisely. And when these genes are mutated, development gets profoundly confused. You might see stages repeating when they shouldn't, or things happening way too early or too late.
Okay, so today we're focusing our deep dive on a specific pivotal mystery involving two key players from this group, the heterochronic genes LIN4 and LIN14.
Yes, the classics.
Genetically, we knew LIN4 was crucial for normal timing, and it seemed to work by somehow, putting the brakes on LIN14. But the big question for a long time was, how?
What was the actual molecular mechanism? How did LIN4 control LIN14?
Exactly. And our map for this exploration, which you shared with us, comes from a foundational research paper that really cracked this molecular puzzle for the first time.
It was a landmark paper.
So the mission of this deep dive, tailored just for you, the listener, is to walk through the key experiments and findings from this source material, understand the surprisingly novel mechanism LIN4 uses,
And it really was surprising.
and appreciate its broader implications for how genes are controlled, maybe even in us. Okay, let's unpack this.
Let's do it.
So let's start maybe by looking at the problem through the lens of genetics, as the source material does. What happens when LIN4 and LIN14 aren't functioning correctly? What do the worms look like?
Right, the phenotypes. The paper describes them clearly. If you had a loss of LIN4 function, meaning LIN4 isn't working, the worm shows retarded development.
Retarded, meaning?
Meaning early larval cell division patterns get inappropriately repeated at later stages, so things that should happen only early on keep happening.
Okay.
These worms might have extra molts, they might fail to develop adult structures like the vulva, which is needed for egg laying, or the adult cuticle. They often can't lay eggs. Essentially, they're stuck repeating juvenile programs.
Got it. And LIN14 mutants show the exact opposite then.
Yes, precisely. A loss of LIN14 function leads to precocious development. Early stages get skipped, and later stage programs kick in way too soon.
So the worm rushes prematurely towards adulthood.
Exactly. Too fast.
And the source points out a crucial genetic relationship here, something called epistasis. This sounds important.
Oh, it's critical. Epistasis tells you how genes interact in a pathway. In this case, if a worm has a loss of function in both LIN4 and LIN14.
Okay, both broken.
Right. The LIN14 null phenotype, that precocious skipping ahead development, it overrides the LIN4 phenotype. So the worm looks like a LIN14 mutant, not a LIN4 mutant.
Okay, wait. So if LIN4 normally stops LIN14 and you break LIN4, LIN14 runs wild, causing the retarded state.
Correct.
But if you break LIN14 itself, it doesn't matter if LIN4 is broken or not, because the thing LIN4 was supposed to stop is already gone.
You've got it. That's the logic. It strongly suggests that LIN4 normally functions upstream of LIN14 in the pathway and acts as a negative regulator. It's the brake on the LIN14 gas pedal.
Okay, that makes sense. A brake and a gas pedal.
Uh-huh.
So the paper then connects this genetic story to the actual LIN14 protein, the product of the LIN14 gene.
Right. What's the protein doing? In normal wild-type worms, LIN14 protein levels are high in late embryos and the very first larval stage, L1. But then
They drop.
They drop dramatically. By the L2 stage, the second larval stage, LIN14 protein is way down.
And that decrease must be important.
Absolutely essential. It's required for the worm to transition properly from L1 specific developmental programs to the next set of programs for L2 and later stages. It's like flipping a developmental switch.
So it makes perfect sense then that in the LIN4 mutants where the LIN14 brake is broken, or in these things called LIN14 gain-of-function mutants where the gas pedal is stuck on.
Right, where LIN14 activity is abnormally high or persistent.
In both cases, the LIN14 protein levels stay abnormally high later in development than they should.
Exactly. That persistent high level of LN14 protein keeps those early L1 developmental programs running inappropriately, explaining that retarded phenotype. The switch never fully flips off.
Okay, so we know LIN4 is the brake, acting somehow to reduce the amount of LNN14 protein at the right time. But how? How does it actually apply that brake? Here's where the source material apparently reveals something really surprising.
Yeah, this is where the picture gets interesting at the molecular level. Yeah. Usually when you see a protein level decrease like that, your first thought might be, well, the cells stop making the blueprint, the mRNA.
Right, transcriptional control.
Yeah.
Less message, less protein.
Exactly. But the paper points out something crucial. The amount of LIN14 mRNA, the direct template for the protein, actually remains relatively constant throughout development. It doesn't drop significantly when the protein does.
Wait, hold on. The recipe, the mRNA, is still there in high amounts, but the final product, the protein, is decreasing.
Precisely. That's a really strong indicator that the control isn't happening at the level of making the mRNA or transcription. It must be happening after the mRNA is made.
Post-transcriptional regulation.
Post-transcriptional regulation. It's likely affecting the efficiency of translation, the process of actually reading the mRNA blueprint to build the LIN14 protein.
Okay, so something is gumming up the protein-making machinery for LIN14 mRNA specifically.
Yeah.
And the researchers in the paper, they zeroed in on a specific part of the LIN14 mRNA itself.
They did. They focused on the 3' untranslated region, or 3UTR. This is a stretch at the tail end of the mRNA molecule that doesn't actually code for the protein sequence.
But it can have regulatory function.
Exactly. It often contains binding sites for regulatory factors. And their experiments, including mapping some of those LIN14 gain-of-function mutations, the ones that escape LIN4's control.
Where do they map?
They mapped right to this 3UTR. This showed that this specific sequence in the LIN14 3UTR is a necessary and sufficient cis-regulatory element.
Cis meaning it's part of the same molecule it's regulating, right?
Right. It's a control sequence physically located on the LIN14 mRNA itself. So to get that normal temporal decrease in LIN14 protein, you need two things. You need functional LIN4 acting in trans from somewhere else.
A brake molecule.
And you need this specific sequence in the LIN14 mRNA's 3UTR acting in cis, the docking site for the brake.
Okay, so this points really strongly to LIN4 working through or somehow interacting with that LIN14 3UTR sequence.
Exactly. The puzzle pieces are fitting together. Now the question is, what is LIN4? What is the molecule that LIN4 produces that interacts with that UTR?
Right. So to answer that, the researchers had to actually find and clone the LIN4 gene itself, physically isolate the DNA.
Which, back then especially, involved some really painstaking work. Techniques like chromosomal walking.
Sounds arduous.
It was, essentially using known genetic landmarks on the worm's chromosome to sort of step your way along the DNA sequence until you zeroed in on the region where genetic mapping told you LIN4 should be, and then transformation rescue.
Transformation rescue, that sounds like the key experiment to confirm you've got the right piece of DNA.
It really is. Once you've isolated a chunk of DNA you suspect contains the gene, you basically inject or introduce it back into a worm that has the mutant phenotype, the retarded development in this case.
Okay.
If that specific piece of DNA rescues the phenotype, meaning the worm now develops normally, well then you've likely found the functional gene sequence.
Makes sense. Prove it works. And the paper describes how they identified a specific, actually pretty small, 693 base pair DNA fragment that could fully rescue the LIN4 mutant phenotype.
Yep, that little fragment contained the functional LIN4 gene.
And there was an interesting detail mentioned in the source material about where this sequence was found.
Yeah, kind of a weird quirk. They found this functional LIN4 sequence was actually located physically within an intron, one of those non-coding regions that get spliced out of another, much larger gene.
So tucked inside another gene.
Right. But crucially, their rescue experiments showed that the small LIN4 fragment worked perfectly fine on its own. It could function independently of that larger host gene it was embedded in.
Okay, so they found the functional DNA sequence for Lin4. The next logical step, you'd think, is to figure out what protein it codes for, right?
Right.
What's the structure? What does it do?
That's what everyone expected. Find the gene, find the protein.
But this is where the paper delivered one of its most groundbreaking findings, and it went completely against all expectations at the time.
It was a huge surprise. The data pointed very strongly to the conclusion that the Lin4 gene, despite being this critical developmental timing regulator, does not appear to encode a protein.
Whoa. Really? A gene that controls development but doesn't make a protein? That must have been, well, quite a shock back then.
It absolutely was. It challenged the central dogma, in a way.
How did they even come to that conclusion? What was the evidence?
They had several compelling lines of evidence described in the paper. First, they did phylogenetic comparison. They got the Lin4 sequence from 4 different but related Caenorhabditis worm species.
Okay, comparing across species.
And they found that the basic nucleic acid sequence, the string of DNA or RNA letters A, C, G, T, U, was highly conserved across these species. That conservation strongly suggests it's functionally important, right? Nature kept it the same.
Makes sense. If it works, don't fix it.
But when they looked for conserved open reading frames, ORFs, the sequences that look like they could actually code for a protein, starting with a start codon, ending with a stop codon.
Right, the protein recipe itself.
They couldn't find any ORFs that were consistently conserved across all 4 species. There were little insertions and deletions in the sequence between species that would completely scramble any potential protein code. They'd cause frame shifts.
Ah, so the underlying ddRNA sequence was kept, but any potential protein coding information within it wasn't. That's weird.
Very weird if you're expecting a protein. That was one major piece of evidence. Second, they performed mutagenesis experiments directly on their rescuing Lin4 DNA fragment.
Meaning they deliberately broke things.
Exactly. They introduced specific mutations designed to break potential protein codes, things like point mutations to create stop codons prematurely, or mutations causing frame shifts, or altering a potential start codon.
Things that should absolutely kill a protein's function.
Absolutely. If Lin4 worked by producing a protein, these mutations should have destroyed its ability to rescue the mutant worm in those transformation assays we talked about.
And did they?
No. None of these mutations, targeted at potential protein coding sequences, significantly disrupted the ability of the DNA fragment to rescue the Lin4 mutant phenotype.
Wow. Okay, so the sequence is conserved, but doesn't look like a typical protein gene across species. And Dabby deliberately breaking potential protein codes within it doesn't stop it from working. That's pretty convincing.
It's very strong evidence against a protein product being the functional output.
So the million-dollar question then, if Lin4 doesn't encode a protein, what does it make? What is the actual functional product?
And that's the revolutionary part, the real punchline of the paper. The researchers hunted for other possible products and discovered that Lin4 encodes not a protein, but small RNA molecules.
Small RNAs.
Yeah.
Not mRNA that codes for protein, just small RNAs that do the job themselves.
Exactly. Final products that are RNA.
How do they even find something potentially so small and maybe not very abundant?
They had to use highly sensitive RNA detection methods. The paper mentions techniques like northern blots and RNase protection assays. These let you specifically look for RNA molecules of certain sequences and sizes within the total RNA extracted from the worms.
And using these techniques,
They found 2 distinct, very small RNA transcripts that originated specifically from that Lin4 DNA region.
and how small are we talking here? Like really small.
Really small. The paper reports a larger transcript, which they called Lin4L, of about 61 nucleotides in length, and a much smaller one, Lin4S, of only about 22 nucleotides.
22 nucleotides. That's tiny. For comparison, a typical mRNA coding for a protein might be hundreds, even thousands of nucleotides long, right?
Easily. So these are genuinely minuscule RNA molecules.
Okay, they found these tiny RNAs, but how did they confirm that these specific small RNAs were the actual functional products, the things responsible for Lin4's activity in regulating development?
They had crucial genetic evidence linking these RNAs directly to Lin4 function. First, they showed these small RNAs were present in RNA, isolated from normal wild-type worms, and they were also present in the Lin4 mutant worms that they had rescued by adding back that functional Lin4 DNA fragment. Makes sense. Put the gene back, you get the product back.
Right. But were they missing in the original mutants?
Critically, yes. These small RNAs were undetectable in the original Lin4 null mutant, the E972 allele mentioned, where the Lin4 gene was known to be completely non-functional. If the gene is broken, the small RNAs aren't made.
Perfect. Gene presence correlates with RNA presence. That's strong. Was there more?
Even more compellingly, they found and characterized a new Lin4 loss-of-function mutation, one called May767. This wasn't a dilution, just a single change in the DNA sequence, a point mutation, a C change to a T.
One letter change breaks the whole gene's function.
Yes. And crucially, they found that this single C to T change altered the sequence within both of these small RNA transcripts. It wasn't somewhere else in the DNA fragment. It was right in the sequence of the small RNAs themselves.
Wow. So a mutation that breaks the gene's function is a mutation in the sequence of the small RNA product that directly links the RNA sequence to the biological function.
Exactly. It's powerful genetic evidence. If you change the RNA sequence, you disrupt the gene's activity. And they even figured out the relationship between the two RNAs.
Did they?
Yeah, based on mapping experiments, their data suggested that the smaller RNA, the 22 nucleotide Lin4S, might actually correspond to the 5' end, the front end, of the larger 61 nucleotide Lin4L transcript. Maybe Lin4L gets processed down to Lin4S.
Okay, so the tiny 22 nucleotide piece, Lin4S, seems like it might be the really key player here.
That's what the evidence started to suggest.
So let's put all these pieces together now. We know Lin4 makes these tiny RNAs, particularly maybe the 22 nucleotide Lin4S. And we knew from earlier that Lin4 acts through the 3-UTR, the Lin14 mRNA, to regulate LAN14 protein levels. How do these two findings connect?
This is the really elegant part, the core mechanism that the paper proposed and provided strong evidence for. They looked closely at the sequence of those Lin4 small RNAs and compared them to the sequence of the Lin14 3-UTR.
Did they find anything?
They found something remarkable. Parts of the Lin4 small RNAs, particularly Lin4S, are complementary to specific sequences found within the Lin14 3-UTR.
Complementary meaning they can base pair, like A with U, G with C.
Exactly, like two sides of a zipper fitting together. The nucleotide sequences can literally stick to each other through hydrogen bonds.
Okay, so the small RNA can physically bind to the target mRNA.
That's what the sequences suggested. In fact, the Lin14 3-UTR contains a specific target sequence element that is repeated 7 times. And the Lin4 RNAs contain sequences that are complementary to this repeated element.
7 potential binding sites.
7 potential docking points. This strongly suggested a direct RNA-RNA interaction, where the Lin4 RNA binds directly to the Lin14 mRNA in those locations within the 3-UTR.
Acting like an antisense molecule, binding to the sense mRNA strand.
Precisely that. Antisense regulation, but mediated by a tiny RNA targeting the 3-UTR.
And the evidence supporting this specific interaction, this base pairing idea, was strong.
Incredibly strong, based on multiple lines described in the paper. First, this sequence complementarity, the potential for Lin4 RNA to bind the Lin14 3-UTR, is conserved between C. elegans and its relative C. briggsii.
So if the mechanism is important, the sequences involved should be conserved too.
Exactly. The regulatory relationship is conserved, and so are the interacting sequences. Second, remember that Mach767 point mutation in Lin4 that broke its function.
The C to T change.
Yeah, that single change. It disrupts a base that is predicted to be directly involved in this base pairing, this complementarity, with the Lin14 3-UTR sequence. If you mess up the pairing, the interaction fails, and the regulation fails.
Okay, that makes perfect sense. And what about the Lin14 side?
Remember those Lin14 gain-of-function mutations? The ones that make Lin14 insensitive to Lin4's breaking effect?
Right, where the protein stays high even when Lin4 is present.
Those mutations map precisely within these Lin4 complementary sites in the Lin14 3-UTR.
Get out. So if you mutate the target site on the Lin14 mRNA, the Lin4 RNA can't bind to it effectively, and regulation fails.
Exactly. You break the docking site, the break molecule can't attach.
Wow, that's really compelling. You have the genetic interaction. You find Lin4 makes small RNAs. Those RNAs have sequences complementary to the target gene's UTR, mutations in the RNA sequence break function, and mutations in the target UTR sequence break regulation. It's a beautiful, complete story of interaction.
It really is. It provides incredibly strong support for the model. Lin4 small RNAs bind directly to the Lin14 mRNA 3-UTR via this specific antisense complementarity.
Okay, so the binding happens. How does that binding actually lead to the decrease in LAN14 protein? What's the consequence of that RNA-RNA hug?
The paper proposes the most likely outcome. The binding of these tiny Lin4 RNAs to the Lin14 mRNA 3-UTR somehow inhibits the translation of that mRNA into LAN14 protein.
So it stops the protein from being made, even though the mRNA recipe is still there.
Exactly. This interaction interferes with the ribosome or translation initiation factors or something in the protein synthesis machinery, preventing it from efficiently reading the LIN14 message and building the protein. This causes the necessary temporal decrease in LAN14 protein levels during development.
And they had a thought about which RNA, the long or short one, was doing the work.
They noted, as we mentioned, that the smaller RNA, LIN4S, the 22 nucleotide one, was significantly more abundant than the longer LIN4L, which suggests that LIN4S might be the primary functional regulator, the main workhorse. And its abundance suggested it might act stoichiometrically. Maybe you need, one or a few LIN4S molecules binding to each LIN14 mRNA molecule to effectively shut down its translation.
This research was truly groundbreaking then, because it described, potentially for the first time, a developmental timing gene being regulated to post-transcriptionally by a small antisense RNA acting through the 3-UTR of its target.
Absolutely. It unveiled what seemed to be a completely novel class of gene regulation mechanism. It wasn't the standard model of protein transcription factors binding DNA or even proteins binding RNA.
This was RNA regulating RNA.
RNA regulating RNA directly to control development. It showed that RNA itself could be a final, functional regulatory product of a gene.
And the exact molecular nitty-gritty of how that RNA-RNA binding actually jams the translation machinery, whether it physically blocks the ribosome or maybe recruits other silencing proteins or destabilizes the mRNA.
Right. Those precise downstream details were still areas for future research. And indeed, a whole field exploded from this discovery trying to figure that out for microRNAs in general. But the core discovery here, the small RNA product of LIN4 acting via antisense pairing to the LIN14 3-UTR to control protein output, that was the monumental finding.
It really did change the game, didn't it? It opened up the idea that there's this whole hidden layer of regulation mediated by tiny RNAs.
Completely changed the way we think about what genes are and what their products can do. Before this, the central dogma was very protein-centric. LIN4 showed that RNA can be the star player, too.
So to sort of wrap this up, we started with this puzzle of developmental timing in a little worm, C. elegans, and how one gene, LIN4, controls another LIN14.
You mean brake and the gas pedal.
Right. And we've seen how this deep dive into the molecular details, guided by the source material, revealed a truly surprising mechanism. LIN4 isn't a protein-coding gene at all.
Nope, no protein.
Instead, it produces these tiny RNA molecules that act as antisense regulators, binding directly to the LIN14 mRNA to dampen down protein production at precisely the right developmental moment.
A remarkably elegant and efficient switch.
This journey really shows us the incredible diversity and elegance of gene regulation in living organisms. Sometimes the most crucial players are small, hidden, and work in completely unexpected ways.
Turning switches on and off to guide these incredibly complex processes like development.
And this deep dive hopefully has given you a shortcut to understanding this really fundamental biological switch.
It highlights that biological systems often find these really creative, maybe unexpected solutions to molecular problems. Who would have guessed, decades ago that a tiny RNA could silence a protein like this?
Absolutely. So here's a final thought for you, the listener, to consider. Given how challenging it actually was to identify LIN4 as a small RNA gene, partly because it was so small, partly because it was hidden within another gene's intron.
Yeah, it wasn't obvious.
How many other essential biological processes, perhaps even in our own human cells, might be controlled by similar, tiny, currently overlooked RNA molecules, performing really crucial regulatory roles that we haven't yet fully imagined?
It's a fascinating question. It suggests the world of gene regulation clearly has many more layers, many more secrets, still waiting to be discovered.
Perhaps hidden in plain sight, just like LIN4 was for a while.
Could be. There's likely still a lot we don't know about how our own genomes work at this level.